Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Central University's Common Entrance Test question paper Biochemistry conducted in the year 7th June 2023. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers, I have uploaded them in my channel. You can refer to my channel playlist for the same. Coming to the first question. The largest membranous cellular organelle, it is nucleus. Answer is option 3. Which of the following cellular organelle is called the digestive tract of the cell? It is naturally the lysosomes. Answer is option 4. Then mitochondria involved in oxidative phosphorylation, beta oxidation, yes. Independent protein synthesis, yes, it has got its own DNA. Ribosomes, so it is involved in independent protein synthesis, so both 1 and 2 are correct. Size exclusion chromatography or gel filtration chromatography separate proteins on the basis of stroke radius. So stroke radius, it refers to the radius of a spherical particle or molecule that has the same properties as the molecule of interest, and it measures it, or I can say it is a measure of the size of the molecule in a solution. That is called as stroke radius. So size exclusion chromatography, it separate based on stroke radius. Then molecular weight and shape, yes, 2 is also correct. So answer here is option 3, that is both 1 and 2. Then next one. In spectrophotometric measurement of proteins, which of the following amino acid contribute the maximum? So for spectrophotometry, there are 3 amino acids that contribute for the maximum measurement of proteins, that is tyrosine, phenylalanine and tryptophan. Answer is option 1. Then one of the following is not related to enzyme-linked immunosorbent assay. Monoclonal and polyclonal antibodies. Antibodies are used in ELISA. Antigen and coupling enzyme. Yes, that is also used. Electrophoresis. Electrophoresis is not conducted in ELISA. Secondary antibody. Secondary antibody is also used. It binds to the primary antibody. Then you add the substrate and the color is produced. So secondary antibody is also used. Then next one. If the molecular weight of protein is 2,20,000 gram per mole, what would be the number of amino acids in the protein, assuming mean molecular weight of an amino acid to be 110 gram per mole? So number of amino acids is equal to molecular weight of protein by mean molecular weight of amino acid. So you will get the answer as, substitute the values and you will get the answer as 2000. Answer is option 2. Then next one. Which of the following statement is incorrect? Ramachandran plot is related to protein structure. Yes, for secondary structure determination you use Ramachandran plot. Watson and Crick discovered structure of DNA, proteins, RNA. No, they discovered only the structure of DNA, not RNA and proteins. So they have asked for the incorrect statement. So answer here is option 2. Okay, next one. Calvin cycle incorporates all the carbon backbone of biomolecules. That is also correct. Then next one. Find the wrong one out. Nucleotide minus phosphoryl group is nucleoside. Yes, that is correct. Purines and pyrimidines are double and single ring compounds respectively. That is also correct. Purines and pyrimidines are linked to sugars by peptidoglycan bond. It is not peptidoglycan bond, it is N-glycosidic bond. So that is your answer. They have asked for the wrong one. Adenosine and uridine are ribonucleosides. That is also correct. Then next one. A phosphodiester bond formed between. Naturally it is between polynucleotides. Answer is option 2. Watson-Crick base pairing is hydrogen bonding in a single-stranded DNA. That is not Watson-Crick base pairing. Non-covalent hydrogen bonding between two DNA strands. Yes, that is correct. That is Watson-Crick base pairing. 2 is correct. Van der Waals forces between bases. Van der Waals forces are weak intermolecular forces that play a role in DNA structure and are not directly involved in Watson-Crick base pairing. Then third one, phosphodiester bonds between nitrogenous bases of opposite strand. It is not opposite strand, but they are covalent bonds that link nucleotides within a strand. And do not connect nitrogenous bases of opposite strand. Then next one. The complementary sequence for the DNA stretch. They have given the sequence would be. So first is G. So complementary to that will be C. Complement to A will be T. T A. Then G. T T A G. C T T G A. First one itself is correct. So answer is option 1. The two common antioxidants that also act as pro-oxidants in biological systems. So antioxidants are those that neutralize free radicals, then prevent or reduce oxidative stress, protect cells, proteins and DNA from damage. Pro-oxidants, they play the exact opposite role of antioxidants. They promote or generate free radicals, then they increase oxidative stress, can cause cellular damage, inflammation and even cell death. So the answer here will be ascorbate and beta carotene. Answer is option 4. Then diabetes is genetic disorder, yes. Metabolic disorder, yes. 3. Genetic and metabolic disorder. That is more correct. So genetic factors include family history, genetic predisposition, etc. And metabolic is factors like insulin resistance, impaired insulin secretion, glucose metabolism, that is imbalance in glucose production, uptake, utilization, obesity and less physical activity are the metabolic disorders. An oxyhemoglobin molecule contains. So oxyhemoglobin, it is a molecule that forms when oxygen binds to hemoglobin, and oxyhemoglobin is responsible for transporting oxygen from the lungs to other body tissues. So an oxyhemoglobin molecule, it contains not carbon dioxide as in one, it contains a heme with iron, oxygen and protein. That is option 4. Then next one. Which of the following mutation is least deleterious in the genome? It is silent mutation. Why? Because silent mutation does not change the amino acid sequence of a protein. So it is less deleterious. Then next one. Given below is the triple nature of codon. The possible permutation of the 4 bases is. So that is naturally given when you learn codons. It is the same thing that they have asked. It is 4 raised to 3, that is 64. 64 can code for 20 amino acids. Then DNA photolysis are enzymes that convert. So DNA photolysis are enzymes involved in repairing DNA damage caused due to UV light. They convert pyrimidine dimers, which are abnormal bonds between two pyrimidine bases, back to their monomers. And they require FADH2 as the cofactor. So first option, if you see, it is pyrimidine dimers to monomers with FADH2 as cofactor. Answer is option 1. What are the possible number of reading frames in a given double-stranded DNA molecule? So there are 3 forward reading frames and 3 reverse reading frames, and each reading frame can encode different protein sequences, and only one or a few of these reading frames encode functional proteins. So the possible number of reading frames is 6, 3 forward and 3 reverse. Then next one. During translation, the directionality of polypeptide synthesis is, it is from N-terminal to C-terminal direction. Answer is option 4. Find the odd one among the following. James Sumner urease, holoenzyme, functional enzyme plus coenzyme or metal ion. Apoenzyme, enzyme plus prosthetic group. Apoenzyme, it is a protein that requires a prosthetic group or a cofactor to become functional enzyme called holoenzyme. Then hexokinase is Mg2 plus dependent. So here urease, holoenzyme, hexokinase, they are all complete enzymes. They are all holoenzymes, but apoenzyme is not. So that is the odd one out. Then next one. The catalytic efficiency of an enzyme refers to Kcat by Km. Answer is option 3. Okay, next question. Km of an enzyme catalyzed reaction is. So it is the substrate concentration at which reaction rate is half of Vmax. So if you see the diagram here, this is the Vmax by 2, that is half of the rate of reaction, half of Vmax. This is the Km. So Km is the substrate concentration. Substrate concentration at which reaction rate is, or rate of reaction is, half of the maximum. So answer is option 1. Identify the wrong statement about penicillin. It is an irreversible inhibitor. Yes, it targets glycopeptide transpeptidase. Yes, in the bacterial cell wall, during bacterial cell wall synthesis. Then it consists of a beta-lactam ring plus proline. It doesn't contain proline, it contains a beta-lactam ring but not a proline. So they have told about the wrong statement. So the answer here is option 3. It forms covalent bond with active site serine. That's also correct. The most abundant enzyme on earth is your ribulose 1,5-biphosphate carboxylase or oxygenase, because plants are more abundant. This enzyme is found even in algae also. Then Gemelin test and Van der Berg reaction are used for. So they are used for identification of bilirubin in urine and serum respectively. Answer is option 4. Then which of the following are involved in buffering our blood system? So it is bicarbonate and phosphate buffers. Answer is option